means nerves and receptors. And some of those receptors you've already covered in skin chapter. And then 14 is autonomic nervous system. And then 15 is special senses.
Okay.
Nervous system is at the center of everything we feel, think, and do. Feel requires receptors and the sensory division of the nervous system. It's the sensory division that brings signals from receptors to processing centers in the brain and spinal cord.
Where's the other one?
And that's where the thinking happens, the processing centers within the brain, within the spinal cord. That's where decisions are made. Consciously or subconsciously. A conscious decision would be you feel thirsty, and so the behavioral response is you seek out water. You're aware of the fact that you're thirsty, right? You're aware of the fact that you're thirsty, and so you purposely find some water and drink it. Subconscious would be you just stood up, you've been lying down for a while, sudden drop in blood pressure upon standing. You're not aware of it unless it makes you feel funny, and for some people it does. But what your receptors, your baroreceptors, are detecting is a sudden drop in blood pressure, and signals go to the brain, and the brain responds, and heart rate goes up, and stroke volume goes up, blood vessels constrict, and very quickly blood pressure goes back to where it's supposed to be. And especially when we're young, it's so quick we don't typically notice that it even happened. So a lot is going on behind the scenes that you're unaware of. And then the doing. That's motor division of the nervous system. So the outcome of the thinking, the processing, is often a response is elicited. Something needs to happen, consciously or subconsciously. So then motor neurons will send signals to effectors. And in general, what are the two main effectors in the body? What are neurons communicating with other than each other? Glands. Could be endocrine glands, could be exocrine glands. Like you smell food and then you start salivating. That's autonomic nervous system stimulating secretion from exocrine glands. Glands or muscles. Like when blood pressure drops, which muscles are stimulated to contract in order to increase blood pressure? Smooth muscles, cardiac muscle. Cardiac muscle is going to contract more often, heart rate goes up, more forcefully, stroke volume goes up. And smooth muscles and blood vessels will contract, the vessels constrict, blood pressure goes up. So for the most part, the targets, the effectors, are glands and muscles. That's what we're controlling. There's some other things too, but that's most of it. For example, adipocytes. That would be a non-gland, non-muscle target. The functional cell of the nervous system is the neuron. Or you could say it's the smallest functional unit of the nervous system. And again, the way you figure out what that is, you look at what the organ or system does, and then what's the smallest thing in that organ or system that does the very same thing. In general, what does the nervous system do? Receives, processes, sends signals. And neurons can do that. The dendrites, the cell bodies, receive signals. Those signals are processed at the cell body. And they may or may not result in excitation of the axon. That is an action potential being generated. if an AP is generated, that neuron is going to send a signal to somebody else. It's going to release neurotransmitter and communicate with another cell. The nervous system includes all nervous tissue in the body, organized along with blood vessels and connective tissue into the following components: the CNS, which is brain and spinal cord, the PNS, which is nerves, and you could also say receptors, because outside of the special senses organs, receptors are nerve endings. Sensory nerve endings form most of our receptors. You saw that in the skin. They came in two flavors: free nerve endings, for like nociceptors, which is pain, thermoreceptors, or encapsulated. Meissner's corpuscle, Ruffini corpuscle, lamellated corpuscle. So unencapsulated or free versus encapsulated. In addition to the central nervous system and the peripheral nervous system, which is chapters 12 and 13, you could also add special senses organs, specialized components of the nervous system. Chapter 14. No, 15. Chapter 15. Nervous system controls, regulates body functions. It is one of the control systems of the 11 organ systems. Nervous and endocrine are the control systems. They regulate the activities of other organs and thus other cells. And typically those other cells are muscle cells or gland cells. Quite often, the nervous system is operating in a negative feedback fashion. And instead of calling them negative feedback loops, which you could, they're typically just called reflexes. The one that regulates blood pressure is called the baroreceptor reflex. But when you map it out, you basically have mapped out a negative feedback loop. You have receptors, you have a control center, you have effectors. You're trying to produce an effect that is opposite of the problem you started with. Right? If you have low blood pressure, baroreceptors detect it, send signals to a part of the brain called the medulla oblongata. And there you have your cardiovascular control centers. Signals are sent out to effectors, heart, blood vessels, causing changes. Heart works faster, stronger, blood vessels constrict. That create the desired effect, increased blood pressure. So here's the basic components of a reflex, which are also the components of a negative feedback loop. A receptor, sensory neurons that take the information from the receptor into the central nervous system. That's where the integration centers are found. What's another name for integration center?
Control center?
The control center. Processing center. It's going to be in the brain or in the spinal cord. That is, it's going to be in the CNS, central nervous system. And if there's a stimulus, then there needs to be a response. So the integration center is going to send signals down motor neurons to the targets. The target cells or effector cells, which is usually glands or muscles. So in the example here, maybe we feel something crawling on our skin. We think it's a bug, so we want to move our hand away. That information goes to the spinal cord. Motor signals are sent back out, and you reflexively move your arm. Not sure if that works with the bug on the skin. Certainly works with pain. The withdrawal reflex from a painful stimulus doesn't even require the brain. In fact, by the time you're aware that you've touched something painful, you've already started to move away from it. Like putting your hand on a hot stove top. By the time you realize what you've done, your hand's already gone. That's because the reflex doesn't even have to go past the spinal cord. That makes it faster. If you send signals up to the brain, processed up there, came back down, it would take longer. So for withdrawal reflexes, we want it to be really quick. But the processing center can be the brain or spinal cord. Organization of the nervous system. So you've got the CNS, you've got the PNS. CNS, brain, spinal cord, PNS, nerves. And the nerve endings of sensory nerve fibers form receptors. And those receptors are going to gather information regarding stimuli from our internal environment, including our internal organs, from skin, tendons, muscles, and also from the external environment. And then the CNS, based on the input, sends signals out through the motor division, motor nerves, to glands or muscle. If it's skeletal muscle, it's the somatic division. So those lower motor neurons from chapter 9 were sometimes called somatic motor neurons. And it's a 2-neuron chain to get from the brain to the skeletal muscle. Upper motor neuron goes from brain down the spinal cord. And in the spinal cord, you have a synapse with the cell body of the lower motor neuron, which goes all the way out to the muscle. 2-neuron chain, upper, lower. If it's anything other than skeletal muscle, then it's controlled by the autonomic division. Smooth muscle, cardiac muscle, endocrine glands, exocrine glands, anything other than skeletal muscle. It's pretty extensive. There's a lot to discuss, so there's a separate chapter dedicated to the autonomic nervous system, chapter 14. And the 2 divisions are parasympathetic and sympathetic. In a nutshell, sympathetic is involved in fight or flight, response to stress. Even exercise is a type of stress. Good stress, but a stress to the body. Parasympathetic is involved in rest and digest, resting conditions. Back to organization, the bottom of page 1 there. So within the brain and spinal cord, you've got processing at different levels. The cerebrum provides higher level processor. Includes the seat of intelligence, so processing intellectual information. That's where intellect is housed. Inhibition, where you don't do certain things even though you may feel like it because you realize what the consequences may be. Right? So that's judgment, inhibition. Language, process language in the brain. The cerebrum is a part of the brain, it's not the whole brain. It's the most highly evolved part of the brain. It's really what separates us from all other animals. The brain stem, on the other hand, is very similar across all animals. Same centers, takes care of the same basic processes, which are survival processes. Regulation of urinary system, digestive system, cardiovascular system. Right? Just kind of regulates the activities of all those internal organs. So you don't have to worry about it. You don't have to think about it. Spinal cord and brainstem provide lower level processing via reflexes, providing subconscious control of body functions, basic body functions. And a lot of what we've learned about the nervous system comes from our study of animal models. We work the same way. Of course, the one thing we can't study in animal models, except maybe apes,
Dolphins.
maybe dolphin, is intellect and language and inhibition and all those things that are processed in the cerebrum, the things that separate us from most other animals. But as far as what's going on in the brainstem, we can get that information from mice and rats. And that's where we get most of our information. As well, not only the nervous system, but other organ systems as well. When I did research at UC Davis, it was rats and mice. But they did others. They did cats, dogs, goats, all sorts of different animals, primate center. But by and large, the most abundantly used animal model, mice and rats. Cheap, easy to maintain, breed like crazy. And nobody seems to mind too much. They get a little sad with dogs and cats, but they don't mind when mice and rats are sacrificed for research. Peripheral nervous system, nerves. Nerves are bundles of axons. So when you see the term nerve fiber, what part of the neuron is it referring to? The axon. Or if you see the term nerve cell, what is it referring to?
Body.
An entire neuron. A neuron is a nerve cell. The axon is a nerve fiber. And muscle fibers and nerve fibers look very similar. They're really, really long and slender and cylindrical. And that's where the term fiber comes from, because in the end, they're cells, right? They're not fibers. So fiber has to do with appearance more than anything. And we throw that term around a lot. We'll talk about sensory fibers and motor fibers. Really, we're talking about axons of sensory neurons, axons of motor neurons. So you take all these nerves in the body, and then you categorize them based on where they come from. If they come from the spinal cord, then they're called spinal nerves. But if they actually come off of the brain, and 12 of them do, they're called cranial nerves. And they have names, and they also have Roman numerals 1 through 12. And both of those we'll cover in chapters 12 and 13. Receptors. They're sensitive to specific changes in the internal or external environment. In other words, they are stimulus specific. A touch receptor will not respond to a change in temperature. A thermoreceptor will not respond to a change in pressure. Each one is specific to a given stimulus. Types of receptors, these are really like group names. The mechanoreceptors, and that includes the baroreceptors, the receptors in some of our large arteries that are detecting changes in blood pressure. They function as mechanoreceptors. And what type of gated ion channel do they use? Mechanically gated ion channels, hence the term mechanoreceptor.
How do mechanical they work?
So they're in the membrane of the nerve ending, the sensory nerve ending. And it looks much like a voltage-gated or ligand-gated ion channel. But what actually causes it to open is distortion of the membrane. Right? From pushing down on it, somehow distorting the membrane here. Stretch or pressure or touch, some kind of physical force. Deforming the membrane that houses the channel actually causes it to open. And that whatever that is, it's called a mechanical stimulus. So there's a whole subgroup of mechanoreceptors called stretch receptors. Because what actually causes the deformation of the membrane is stretching. That's what you have in the walls of large arteries. Blood pressure goes up, you stretch the wall. That stretch activates the mechanoreceptors. Right? That's very different than the ones in the skin that respond to touch. You don't have to stretch anything in that case. Just a little bit of pressure. Chemoreceptors.
Chemical.
An example that we saw in chapter 9 that would be considered a chemoreceptor.
ACH?
ACH receptor is a chemoreceptor, a receptor that responds to a chemical stimulus. So some chemical is going to bind to the chemoreceptor to stimulate it. So you have chemoreceptors that are sensitive to potassium, some that are sensitive to hydrogen. What would you call the one that is detecting changes in hydrogen ion levels? pH receptor. Yeah. Nociceptors. Any noxious stimulus will activate a nociceptor. A noxious stimulus is something damaging and thus painful. Extreme acidity or alkalinity. Extreme temperatures, extreme cold, extreme heat. Physical trauma, cutting, tearing, scraping the skin. That's all noxious stimuli. And thermoreceptors, temperature receptors. Some receptors are free nerve endings, while others are receptor cells. The most complex receptors are the special senses organs. I can only think of one example that's not special senses organ, where the receptor is a separate cell. And we saw that in the skin. Stratum basal, purple cells. Had a yellow nerve ending connected to it. What were they called? Merkel cells. So there, it's not just a nerve ending that forms the receptor, it's a separate cell with the nerve ending. And that's not common. I don't know of any other examples except that one. So otherwise, it's just the nerve ending itself forms the receptor, encapsulated or unencapsulated. Special senses organs, at least one separate cell is involved, sometimes multiple cells. So those are much more complex receptors. Sensory division of the nervous system has two functional subdivisions, somatic and visceral. And this is where the term afferent and the term efferent come from. Sensory and afferent both imply the same thing, which is sending signals to the brain. So when you map out a negative feedback loop that involves the nervous system, you could say that the pathway from receptor to control center is afferent. And then efferent and motor mean the same thing, away from the brain. From brain or spinal cord, away from the CNS, to the target, muscle or gland. So on the negative feedback loop, from control center to effector, you could say that's the efferent pathway, EFF. And then you've got somatic versus visceral. Somatic refers to information that you become aware of, which includes most everything in the skin. You're aware of the touch and the pressure, pain, temperature. Visceral afferents, on the other hand, they take signals from internal receptors. They're called interoceptors. And send that information to the brain, and you never become aware of it. So all sorts of things are being detected in our visceral organs, really without our awareness. What's going on in the urinary system, in the digestive system, the cardiovascular system. We're only really aware that something's going on when it is painful. Extreme pressure in the gut or extreme pressure in the bladder. There are some things that we have to become aware of to make a conscious decision to defecate or urinate, for example. But for the most part, we're largely unaware of what's going on in the viscera. Right, so the viscera or the visceral organs, that refers to the internal organs. The organs in your ventral body cavity. That's the visceral. Motor division, 2 subdivisions. Almost the same names. Somatic, and then instead of visceral, autonomic. And sometimes visceral is used, but not a common term. Usually autonomic. And with autonomic, think automatic. Signals that are being sent automatically without your awareness that anything needs to be done, without any voluntary control. Right? Regulating cardiac muscle, smooth muscle, endocrine glands, exocrine glands. Again, the only thing somatic nervous system controls the skeletal muscle. Now, just because you have voluntary, conscious control over skeletal muscle doesn't mean that's the only way it can be stimulated. There are many examples of reflexes that contract skeletal muscles without voluntary intervention. One would be if you were just sitting there, standing there, looking at something in the distance in front of you, and out of your peripheral vision, you see something coming at you. Maybe a ball coming at your head. What do? You duck. If you had to think about it first, process it, it could be a ball, I better... Too late, you've already been hit. And
people do that sometimes.
And there's an example for me that really stands out to this day, and it happened when I was really young. I was on a playground running really fast, looking at something over there, as kids do, running that way. And you know the bars, there's one at this height, and then one at this height, and one at that. So I was like running right for it, and then I just, as I was turning out of the corner of my eye, there's a bar like right there, and I missed it. It was awesome. Went right under it, you know, but obviously it happened so quick, there was no way for me to process that and make that conscious decision, I better duck. I just did. And I was like, whoa, I almost hurt myself bad. And pain withdrawal reflex is another good example. Putting your hand on the hot stovetop comes off before you even realize what's happening. All right, so that's it for the introduction. A lot of that you're going to see again in chapter 12. Chapter 13 as well. Especially this image here. For the nervous tissue, since the big focus, especially in terms of physiology, is the neuron, we'll just get the neuroglia out of the way first. And then the rest of the study guide is going to be focused on neurons. Nervous tissue is more than 80% cellular. So you could say it has pretty high cellularity, more so than connective tissue, not nearly as much as epithelial tissue. 2 cell types, neurons and the cells that support neurons. And their name collectively is glial cells, or neuroglial cells. If you take off the L at the end of glial or neuroglial, that is the name as well. Glia. Glia refers to all the glial cells, or neuroglia. You don't necessarily have to say neuroglial cells. 6 types of neuroglia. 2 that you find in the PNS, 4 in the CNS. In the CNS, the ratio is 10 to 1. And that adds up to about half of the brain's mass. Half of the mass is glia. In general, they're smaller in size compared to the neurons. Darker staining nuclei compared to the neurons. They include astrocytes. And there's a lot of functions, even more than what I have on the study guide. Largest and most abundant. They reach out and they make connections with blood vessels and neurons. And what enables them to do this and form this framework, this branching framework throughout the tissue, is an extensive cytoskeleton on the inside. So they make all sorts of connections, not only with vessels and axons, but with each other. They form a framework by connecting to each other as well. They make direct exchanges between neurons and the blood. So they're helping to regulate exactly what the neurons are exposed to. And there's potentially lots of things in the blood that we don't want to expose the neurons to. Because unfortunately, when it comes to neurons, when those cells are destroyed, they're not repaired. Once they're gone, they're gone. There's very few areas of the nervous system where that's not the case. Very few. Hippocampus, neurons can be regenerated and new circuits form all the time. Olfactory epithelium, those cells can be damaged and repaired. And Jane, was it you that sent me that link about the article where they were taking cells and basically trying to form cells that would regenerate neurons and then put those cells in a transected spinal cord, someone who's paralyzed, to try to reconnect the broken ends so that the person can regain feeling and control in the lower limbs, upper limbs. Did you see where they were pulling those cells from?
I think it was the nasal.
Olfactory cells, yeah. Because of that quality, that characteristic, because they do regenerate. And there aren't too many areas of the brain where we see that. So the olfactory cells that are in the olfactory epithelium send axons that go up through the cribriform plate, the olfactory foramina, and then they synapse with cells in the olfactory bulbs. And that's what they were taking olfactory bulbs, basically, and then culturing cells out of there. Astrocytes contribute to the blood-brain barrier, the BBB. It is what it says it is, a barrier between nervous tissue and blood. Again, because there's things in blood we don't want to expose to neurons, including toxins, viruses, bacteria. Astrocytes do this by surrounding the capillary, and you can see from both images that they send processes out that surround the capillary wall. So if you're going to go through the capillary, you're going to go through the astrocytes. Right? They're going to really regulate exactly what gets to move in and out of those capillaries. And they also appear to release chemical signals that cause the endothelial cells to form more than normal, more tight junctions, more than we see in other capillaries in the body. Because a typical capillary is a place where free exchange is supposed to occur, where most anything can move across that membrane. Because really what we need to move are waste products and nutrients and chemical signals, like hormones. But here we want to limit those things to just the nutrients and the wastes, maybe certain chemical signals, nothing else. So you do that partly by surrounding the vessel wall and partly by making that vessel less permeable than it normally is in other places.
Is the FA, is that the astrocyte?
Yeah.
And what
There's different types, which you don't need to know. But if you look up FA in the histoatlas, it says fibrous astrocyte, which has to do with the typical ones we're talking about in the brain that form this network. Kind of like connective tissue fibrous network. But they're not fibers, of course. But they kind of form a fibrous network with all of their long processes. They guide neuronal migration during development, which means they help neurons make the appropriate connections with other neurons or target cells, muscle cells, gland cells. They secrete chemicals that enhance or suppress synaptic communication. And this is something that we didn't think was the case until recently. If you can enhance or suppress synaptic communication, then you are involved in processing. And we thought that the glial cells were never involved in any type of processing, that it was just neurons, purely neurons. We've even found that some glial cells can conduct potentials, local currents. So it seems more and more that they might be more involved in the actual processing of information than we used to think. They help maintain normal ECF composition, namely potassium. What was one of the causes of physiological fatigue within skeletal muscle? Especially prolonged exercise. Had to do with potassium.
Not enough potassium inside.
Potassium accumulating in the cell or outside? Outside in the ECF. Presumably because pumps aren't keeping up with the potassium going out for repolarization. Pumps aren't bringing it back in fast enough. And then what starts to happen if you play with potassium concentrations between inside and outside? You change the electrochemical gradient. Right? What happens to the concentration gradient for potassium, which is normally outward directed? If you suddenly let the outside concentration go up as the inside goes down. So if normal potassium concentration is this, and then all of a sudden it starts to accumulate in the ECF, does that get shorter or longer? Shorter. If you let the number out here go up and the number in here go down, you're going to lose that concentration gradient. If you let the number out here go up until it reaches what's in here, what happens to your concentration gradient? There is none. Right? If you have equal concentrations, there is no gradient. Now there's lots of depolarization and repolarization going on over and over again with neurons, right? Just like in skeletal muscle cells. More so than skeletal muscle cells, because neurons are always active. So it makes sense that there are cells or some mechanism there to mop up any excess potassium and return it to the cell. And they seem to help in that process. They are even involved in recycling some neurotransmitters, inactivating and recycling some neurotransmitters. Right, so obviously, in a very general sense, they support neurons, but they do so in many different ways. And that includes regulating the environment around the neuron. Levels of neurotransmitter, levels of ions. Even different glial cells, not the astrocytes, but the microglial cells. You have your own phagocytes right there on the premises. You don't rely on the typical immune system, what we call the blood-borne immune system, right? Because what are the immune cells in general? White blood cells, right? Blood-borne cells. Why do you think blood-borne immune cells, your typical white blood cells, are not involved in protecting nervous tissue? And we're talking brain spinal cord.
Because there's not that much.
Could they carry some kind of
Why wouldn't we want them to, but physically, why can't they?
Because of the it's not.
Blood-brain barrier. They can't get across the capillary wall. It's much less permeable than in most places in the body, where they can squeeze through endothelial cells. Here they can't because of all the tight junctions. So if you're not going to use the body's main immune system, then you've got to have your own. And that's what the microglia do. And the microglia, the smallest and least abundant type of glial cell, is a relative of macrophages. And like macrophages, they're able to migrate through the tissue. They're mobile. They're phagocytic. If there is damage to cells, they can remove cell debris. They can remove wastes so they don't accumulate. Obviously, pathogens if present, which is their main job. And we need to have them because there's no blood-borne immune cells in nervous tissue. They can't get through the capillary wall because of the blood-brain barrier. Third one is ependymal cells. They're very similar to epithelial cells. Epithelial cells join together to form an epithelium, a sheet. Ependymal cells join together to form ependyma. Ependyma is a sheet of cells, like epithelium. Many of these cells are ciliated, help circulate the cerebrospinal fluid. They modify the fluid. So what you have down here, and we'll look at this in more detail in chapter 12, are capillaries called choroid plexus. And the filtrate that is produced across the walls of these capillaries is CSF. CSF is capillary filtrate. It is made constantly, and it's resorbed constantly. It's being made, it's being removed at basically the same rate. So there's a high turnover rate to keep this fluid fresh, so to speak. Has the nutrients it needs, waste product levels low. So the fluid, in order to move, or anything that's in the fluid, in order to move between the CSF and the brain tissue, it would have to go through ependymal cells. So they have a say in what gets transported across the ependyma. And then because they surround the capillaries, they also help determine that initial composition of CSF as it's made. As you take capillary filtrate and make CSF, they might prevent certain things from getting into it. So a real picture of an ependyma is shown here from the histoatlas. This is the layer. It looks almost like a simple cuboidal epithelium. And then these little hair-like extensions, that's cilia. And the V here is for ventricle. In the brain, you have a system of ventricles, which are big open spaces. If you were to take a brain and then fill those ventricles with latex, let it harden, and then remove all the brain tissue, so you get this. So what's a solid structure here is actually fluid in the brain. But this is a model of the ventricular system. You have the big lateral ventricles, one in each of the cerebral hemispheres, and a third ventricle, and a fourth ventricle down here. So we're looking at the layer of cells that lines these ventricles here. Choroid plexus, ependymal cell, ventricle, cilia, ependyma.
So the cerebral, the
CSF.
fluid, yeah. The CSF is only in that area, in the ventricle.
It's in the middle of the brain, in the
And down
ventricles.
the
It's in the middle of the spinal cord, in an opening called the central canal that goes all the way down. And it's around the spinal cord and around the brain in the subarachnoid space. So there's little pathways for the fluid to get from the ventricles down the bottom into the central canal, and then out through these little apertures into the subarachnoid space around the brain and around the spinal cord. So the brain and spinal cord literally float in water, in CSF. And that reduces the weight by 97%. Because the weight of the brain and spinal cord would actually crush the tissue because it's so delicate. But there's fluid inside as well, not just around brain and spinal cord, but inside.
Can you please repeat the CP and the C?
C cilia, CP is choroid plexus. And if you're ever curious, all of these are in the histoatlas. It tells you what they all stand for.
The CSF, does it circulate like nutrients too? Or?
It does, yep. It's kind of like synovial fluid in a synovial joint. Fourth one is oligodendrocytes. These are the ones we saw in the quiz in your pre-lab assignment. Processes wrap around axons to form myelin sheath. You form insulation to speed up conduction, faster propagation of action potentials. The processes of one oligodendrocyte can wrap around up to 50 different neurons. And of course, specifically, not the neuron as a whole, but the axons of 50 different neurons. Three here, up to 50. But keep in mind, in doing so, it's just a small segment. If it's 3 or 50, it's just a small segment on each one. And then another oligodendrocyte is going to form the next internode, and another oligodendrocyte, the next one. It's going to take tons of oligodendrocytes to myelinate an entire axon. But each one can form internodes on up to 50 axons. So what's the counterpart of the oligodendrocyte in the peripheral nervous system?
Swan.
The Schwann cell. They have the same function. This figure I got from a different textbook. It shows all of them. And it breaks up the tissue of the central nervous system into gray matter and white matter. And we'll cover this in chapter 12. The difference is gray matter is mostly cell bodies. White matter is mostly axons. And because most of the axons are myelinated, it gives white matter the white appearance. It's all the lipid. Gray matter is gray simply because it lacks myelin, because it's cell bodies and dendrites on the cell bodies, but not axons. And the axons that we tend to find in the gray matter tend to be unmyelinated. So it's just lack of myelin that makes gray matter gray. So you see cell bodies of neurons in the gray matter. The axons with myelin passing through the white matter. Here's a capillary, blood-brain barrier, partly formed by astrocyte processes. Little tiny microglial cells moving around. Ependymal cells. Here's a real image from a similar location. If we're cutting through central canal, then this is actually a cut through the spinal cord. And the brain and the spinal cord both have gray matter and white matter. So here's the real thing. Central canal in the spinal cord. Ependyma around it. So that's right here. This is central canal. And then this is what looks like simple cuboidal epithelium. That's ependyma lining the central canal. Two more glial cells. Satellite and Schwann, and these guys are in the peripheral nervous system. This is a better depiction of the Schwann cell. Here's the myelin wrapped around the axon that's been removed from the image. And then all the cytoplasm, along with the nucleus, gets pushed to the outside. It'd be sort of like taking a tube of toothpaste that just has a little bit left, and you just roll up from the bottom, roll really, really tight, and get everything pushed to the outside. That's what happens as this thing wraps around tighter and tighter and tighter. Satellite cells are called satellite cells because they sort of orbit the cell bodies of neurons in the PNS. And a group of cell bodies in the PNS is called a ganglion. Plural is ganglia. So as I mentioned earlier, a typical sample to show lots of satellite cells will come from the dorsal root ganglion. And that's what you're looking at here. The left, top left and bottom left are both from dorsal root ganglion. In most of these, we actually cut through the cell body of the neuron, and then you can see the satellite cells around it. In this case, we didn't cut through the neuron cell body at all. And so you can actually see the satellite cells covering the top of it. This is the top of the cell body, all satellite cells, solid cluster around it. And then same thing down here. Cross sections through the cell body, satellite cells around it. They seem to have some of the same functions as astrocytes in the CNS. And that means regulating the environment around the neuron, making sure ion levels are appropriate. Perhaps inactivating, recycling some neurotransmitters. And then the Schwann cells, they're surrounding the entire axon. And Guido, this is what I think you were referring to earlier about two axons. If the cell body is in the middle, when you look closely, it's not quite in the middle of the axon. The axon is here, but really close to the cell body, it splits and runs in two directions.
When I saw one in the book, it was in the middle, just in the middle of the nerve fiber.
Oh, a bipolar neuron, yes. The bipolar neuron, one side is dendrite and the other side is axon. In the unipolar neuron, which we have here, it's all axon.
There's another pseudo.
Yeah, there's
Pseudo.
like two or three more.
Yeah, and there's no difference between unipolar and pseudo-unipolar.
Just looks different.
They're the same thing, and I have no idea why they have two different names out there. All right, moving on to the neuron. Neurons have large cell bodies with varying numbers and lengths of processors. The cell body can have so many dendrites with so much branching. It can look like a huge tree or bush coming off of the cell body for a huge surface area. That's the whole reason for having dendrites and branches off of the dendrites. The more extensive they are, the more communication you're receiving from more other neurons. So if you're only communicating with a dozen neurons, you don't need a huge receptive membrane. But if you're communicating with hundreds or thousands of neurons, you're going to need much more surface area to form those synapses. These are branches. These are the actual dendrites. So this multipolar neuron has more than two processes. It's got an axon. Everybody gets one of these. And then a lot of dendrites. 1, 2, 3, 4, 5, 6, 7, 8, 9. I guess we'll count that little guy. 10. 10 dendrites. Most of which branch. And this triangular region at the beginning of the axon is axon hillock. That's where you find that first batch of voltage-gated sodium and potassium channels. So once the local potential, the graded potential, reaches the axon hillock, now you've got voltage-gated channels to generate an AP. Everywhere else on the cell body and the dendrites, ligand-gated ion channels. Voltage-gated are only on the axon. Cell membranes conduct electrical signals. The reason it's plural, because I realized a single cell has a single cell membrane. Regions. You've got the receptive membrane. You have the conducting membrane. Regionally, they are different membranes. They're structurally and functionally different. The membrane of the axon, which has a special name, doesn't have these ligand-gated ion channels. Has voltage-gated, and because of that, generates and conducts action potentials. And what is the special name for the membrane of the axon? Axolemma. So you have plasmalemma, sarcolemma, axolemma. Lemma is sheath. Sheath or membrane of the axon. Axolemma is different from receptive membrane, which is what you find on the cell body and the dendrites. There you have receptors for neurotransmitters. And those receptors are part of ligand-gated ion channels. General characteristics. The neuron can function for life. It's meant to. Much like cardiomyocytes. Meant to function for life. No repair capabilities. When the cell is damaged, and well, I shouldn't say no repair, no regenerative capabilities. When it's damaged beyond repair, it's just destroyed. Becomes cellular debris, cleaned up by microglia. Most are amitotic, which again indicates they're meant to function for life. If they're amitotic, if they don't divide, they don't regenerate. There's no turnover. Now, of course, you got to turn over proteins and membrane and maintain the organelles within a cell. So you have the ability to maintain yourself. Just don't have the ability to regenerate and make new cells. High metabolic rate. Skeletal muscle cells, on average, have a high metabolic rate as well. And because of that, what do they have a lot of? A lot of mitochondria, and they get a lot of blood flow. And there is extensive blood flow throughout the brain and spinal cord. Anybody go to Body Worlds or Bodies Revealed when it was in Sacramento? Did you see the specimens when it was just the blood vessels? Nothing else?
Yeah. Oh my God.
See how extensive? And that was just arteries. That wasn't capillaries, that wasn't veins, that was just arteries.
Yeah, the vein, big one, yeah.
And they do that for just the brain. Kind of looks like a model of a brain made out of blood vessels, but that's how extensive
Very
blood
dense.
flow is.
Yeah, that's fine.
Very dense. Tons of capillaries, tons of blood vessels. So neurons have to have a constant supply of oxygen and fuels to make ATP aerobically. Cell structure. You got the cell body, that's the soma. The soma is typically quite large. Here's a view at 2000 magnification on the left. Everything else coming off of it are processes. One of those, hard to say which one, is an axon. The rest are dendrites. Functionally, it's the biosynthetic center of a neuron. If that is where the nucleus is located and the RER, then what's going on there?
Making stuff.
The control center of the cell.
Well, what do you make?
Right.
What do you use the nucleus and ribosomes to make? Proteins. And a lot of the neurotransmitters that are used are peptides, proteins, amines, amino acid derivatives. Within the nucleus, you have nucleoli, and quite often, plural, nucleoli. And if that's the case, then that means you're making lots of ribosomes. And if that's the case, that means you're carrying out lots of protein synthesis. And it's not just to make